Thank you for this question. I had issues, but they are more psychological than practical. I actually moved the year before Brexit kicked in. I have to say that I've been quite shocked by the Brexit decision, and as several other people, both in Europe but also in the UK. In practical terms, I've not suffered much because there's always been the possibility for UK scientists to access the European funds, and still there is this possibility. So when I moved, I've been awarded, I was awarded an ERC Advanced Investigator Grant, and I had no problems in moving and using it in the UK. And then in the meantime, I've also applied, when it finished, I applied for a new large investigator grant from the ERC program, and in this has been supplemented by the UK government after the award. So basically, no impact on science, no impact also on people, because several of the people who decided to join my laboratory could do this. There is, however, a lot of problems now in other laboratories, especially for the visa system and for the requirement that salaries exceeds a certain threshold to have the visa permit to work in the UK. But more than this, which can be circumvented, there are the same regulations are also in place in the United States. So it's nothing new, but it is the general perception that not necessarily working in the UK is so welcoming as it used to be. And I think that in the perception in European countries, not only in Italy, but I'm seeing also in Germany, in France, is that the UK is no longer a place where moving to is so attractive, which is largely wrong, because the UK universities, the academic atmosphere is very, very international. The King's College London, where I am, it is really multicultural, multi-ethnic, very welcoming, very engaging, very protective also of these equalities. But the perception remains. It is fueled by a lot of press, also the press for the lay public, so general journals, and this is something that hopefully now can be somehow reversed by the change in line of the new government. Oh, they certainly have. I mean, the main reason of morbidities of disease and mortality due to pollution is cardiovascular. One would expect that if the atmosphere is polluted, one would die of respiratory disease. Instead, the person dies because of atherosclerosis and complication in the cardiovascular system. But having said this, I think that the problem of cardiovascular disease is simply aging. So the problem is that in all societies, including in low and middle income countries, people die more now of cardiovascular disease, meaning with these terms, myocardial infarction, so heart attack and stroke, all consequences of atherosclerosis, die more of this condition than any infectious disease put together. So medicine in the last century was a medicine of lung infections, of gut infections, of tuberculosis. People were dying of these conditions. People in this century dies of degenerative conditions, of which cardiovascular disease is the leading cause. I have a very good friend and collaborator in Harvard who sometimes starts his presentation for the lay public asking people in the audience to see who's sitting on his left side, who's sitting on the right side, and deciding who of the three will die because either infarction or stroke. But this is the reality. 30% of humanity died because of cardiovascular disease, and the vast part of this it is because on the one hand there's been an improvement in care, in medications, or people with this condition, but on the other hand, the prevailing parameters has been an increase in life expectancy. So now a person in our country, a man, lives more than 80 years, a woman more than 85 years, and the incidence of this condition is huge. No, certainly. I mean, the cardiovascular disease is among the big culprits of the SARS-CoV-2 disease in general, not because of a direct infection of the heart, which is very, very rare, but because SARS-CoV-2 is a systemic infection that causes inflammation, and this hyperinflammation has a strong consequence on vessels, and so including vessels of the coronary arteries, and so it has its influence in heart disease. There is evidence also that in atherosclerotic plaques there can be presence of the virus, and not because the virus causes the disease itself, because it is there are just traces, but because these traces can stimulate inflammation, and inflammation is a main culprit of all cardiovascular conditions. And so certainly the cardiovascular system is at the center also of the SARS-CoV-2 pandemic. There is still room for improvement, but I think that the situation has changed really drastically compared to 20 years ago. There is a strong awareness of the importance of testing both sexes in all kind of experimentation. This is in the UK. There is almost an obsessive attention to this. All the granting agencies don't award grants for research if there is not a proper consideration of this gender issue. Animal work is not supported if there is not the explicit will to test effect of a treatment, for example, in both male and females. So I think that the situation has really improved drastically compared to only a couple of decades ago. There is obviously still room for improvement, especially in the understanding of the biological reasons why there is a difference between males and females, because it's not only a matter of testing, but also understanding why there are differences in terms of response to drugs, for example. And this is something very, very, very interesting also from the discovery point of view. Here I have to say that I'm really biased, because this is exactly what we do in the laboratory. It will be a tremendous game changer in all medical conditions. The possibility of using RNAs as a real new set of medicines. RNAs means both messenger RNAs, so RNAs that contain the information to produce proteins, but also small RNAs that don't code for proteins, but have a regulatory function. Let's not forget that there are already 18 drugs that are based on small RNAs that have been approved by the Food and Drug Administration and the European Medicines Agency here in Europe, and there will be more and more to come. And as you anticipated, the challenge and the enormous opportunity is now to move from vaccines. So let's remember, obviously, the huge success of vaccines against SARS-CoV-2, but also the new vaccines against cancer. So the RNA technology permitted to develop very quickly vaccines based on RNAs, in which the RNAs have exactly the same sequence as the proteins, those coding for the proteins that are mutated in each individual patient. So starting from the RNA sequence of the tumor, one can develop the personalized vaccine. As there are already preliminary, there is already very preliminary evidence of success for pancreatic cancer, for melanoma, metastatic melanoma, so cancer that were considered untreatable until a few years ago. But the challenge is to go beyond vaccination and use RNA therapies for conditions for which there is not a therapy. What we do in the laboratory is to use both small RNAs and also messenger and larger RNAs to stimulate regeneration of the heart after a heart attack. I mean, the big problem of the heart is that it is like the brain. It is an organ in which cells don't proliferate in the adult life. And so once cardiomyocytes, so the contractile cells in the heart are lost, for example, because of a myocardial infarction or a heart attack, they are not replaced. Well, through mRNA therapies, we can stimulate the regeneration of these cells and so cure the heart. And there is a lot of excitement in this area, as there is a lot of excitement using RNAs to administer to the heart and also to other organs the components of the gene editing system that permits to correct directly mutations in DNA to cure inherited diseases and so on. So my prediction is that in the next 20 years, we will see a flurry of RNA-based medicines in all sectors of medicine. Yeah, organoids are of interest in virtually all fields of medicine, because they are three-dimensional structures that somehow resemble the structure of the organ where they come from, or the organ that are formed of the cells, the main cells where they derive from. So we can have organoids for the gut, which were the first to be developed, but also organoids from the brain, which are formed by brain cells, so neurons and other cell types, organoids for the pancreas, organoids for the kidney, and we can have organoids for the heart. So basically, the heart has 5 main cell components, of which the most important, or is the most relevant in terms of structure, are cardiomyocytes, so the cells that contract every heartbeat. And then there are some support cells, which called cardiac fibroblasts, some immune cells, and the cells that line the vessels. And you can put together all these cell types, and they have the capacity of assembling into three-dimensional structure that, under certain condition, can start contracting rhythmically, as the heart does, because this is an intrinsic property of this organ. And the advantage of these organoids is that you can derive them from the cells of a given individual with its own genetic makeup, including some of the mutations. And this can be obtained by a method that won the Nobel Prize to Shinya Yamanaka a few years ago, that consists in taking any cell from a patient, for example, a cell from the skin or a cell from the blood, inserting just 4 genes. These genes transform these cells into an embryonic stem cell, and from these embryonic stem cells, one can derive the generation of cell of a specific organ, including cardiomyocytes. So this means that one can build, starting from these cells, an organoid This is a sort of mini heart, but with the same genetic makeup of the person who donated the cell from the skin or the cell from the blood. And these organoids can be kept in culture. One can test the effect of drugs on these organoids. One can test new therapies to have a formation of new cells through a sort of regeneration and so on and so forth. So all the advanced laboratories make an extended use of these organoids as a sort of surrogates for the real human heart. We lead from the assumption that the big problem in cardiology is the loss of cardiomyocyte throughout life. So before birth and immediately at the moment of birth, the heart is an organ that has plenty of cells that proliferate and regenerate. If one, for example, damages the heart in the first couple of days after birth, there is complete regeneration. This is proven in mice, but also in pigs. There is even anecdotal evidence that human newborn babies, if they have damaged the heart, the repair is through complete regeneration. It's like a superficial wound in the skin, which after a few weeks is completely repaired without forming a scar. If one waits 1 week, then instead the only way of repairing is through formation of a scar. The reason why this happens is unknown. We and several others are trying to understand, but something occurs in proximity to birth, so immediately after birth, by which the cells in the heart lose their capacity of proliferation. And so basically the main topic we are working on is, on the one hand, to find treatments that block death of the cells. So our prototype drugs in this area is a treatment that can be injected in a vein of a patient with a heart attack, ideally in the ambulance immediately after diagnosis, and somehow freezes the cells in the heart and prevent them from dying. So a cardioprotective therapy that works in the first 2-3 days after a heart attack, while the patient reaches the hospital, is revascularized and is stabilized. And then the other big treatment, as I mentioned before, is a treatment for regeneration. If cardioprotection fails, then we have to do regeneration. This is an area that has been very controversial for very many years, because at a certain point there was the belief, which is wrong, essentially proved to be wrong, that there are stem cells that can be used to regenerate the heart. In reality, the best way of regenerating the heart is to stimulate the proliferation of the survived cells. So bringing the heart back to a condition as it was immediately after birth. So in the first 1-2 days after birth, when regeneration occurs, and so reproducing this genetic program. And so through these RNA technologies, we can do this now. And so basically we can think of achieving cardiac regeneration in addition to cardiac protection. That's a very good question. The question could be rephrased. Since we are working on regeneration, could regeneration also prolong life and so be important for longevity? And the answer is probably yes, because longevity is somehow linked to the capacity of the various organs to heal the damage they suffer in adult life. And so if we are able to heal the heart, heal the brain, heal the blood vessels, heal the kidney, then automatically we would live longer. And so I'm certain in the sense that several of these regenerative therapies, including the one for us in the heart, is the therapy that eventually will impact on longevity. Then longevity has another dimension because this is a longevity by which one can extend life instead of 85 years, can be 87, can be 90, can be 95, but we all have a limit which is biological 120 years. And in that limit is really not understood. So there is no one in the world who can answer a very simple question that is why a mouse, which shares the vast proportion of genes with humans, lives a maximum of 2 years, and a human 120 years. Well, why such a huge difference when we have more or less the same genetic composition? This is an answer that remains unanswered. We know that there are genes that control also the lifespan in these terms. There are fly mutants or worm mutants that can live 4 or 5 times. For us, it would be like living 480 years, for example. But how this can be applied to humans and how to transform a mouse into a living being that lives 120 years, this is a big mystery. And I don't think that this is only related to, going back to the question, to the problem of regeneration of the organs, but it is something more intrinsic to the genetic programs of the organism. Communicating science is not a matter at which every scientist is good, and is not a matter at which every journalist is good. So it requires professional persons who are skilled in both science and the communication of science. The problem is quite variegate, and because the number of such professional people in communicating science is not very high, and several times we see also in the big media, the big journals on the national televisions. We see that generic journalists try to communicate science with disastrous consequences, in the sense that very relevant new pieces of scientific evidence are portrayed as the cure for an incurable disease. We have, if you look at the big titles in the big Italian media, the cure for cancer has already been achieved at least 400 times, I think, and instead we are where we are. And the same for the HIV vaccine, and the same basically in any area. So there is the risk, there is the problem, not the risk, that on the one hand, very good scientists lack the capacity for communication or the interest for communication, which is fine because their job is different. But the institution where they are, they don't have a communication office that works well. On the other hand, the reporting news about science somehow is delegated to journalists who don't understand at all about science, and this is equally disastrous. Oh, I think from the school, from the primary education in school, there is no other way that instill education in science in the school, and with education on scientific topics comes also the need for a continuous education after they leave school or they leave university, because science is evolving so rapidly that unless one has the capacity and feels the need to educate themselves continuously, then the knowledge acquired becomes obsolete. But this mindset must come from the school. And then the one big problem is that our politicians are really very ignorant about science, and what is even worse, they are uninterested about science. When I was the director of the ICGB, the International Center for Genetic Engineering and Biotechnology in Trieste, I started a program that we called Science and the City, in which for several years we brought eminent scientists in a theater in the center of Trieste to dialogue on important topics like genetically modified organisms, stem cells, the vaccines, including topics that were at a certain point of or under the radar, like the stamina problem and the like. And the theater was really crowded, packed with the lay citizens that wanted to know about science, but not in a single time out of 40 or events that I organize, I've seen any political person sitting in the audience, despite many invitations. And that's a huge problem. It is a problem by which in our parliament and our politicians, our administrators don't have the instruments to react to scientific news, and sometimes they follow types of reasoning that is more demagogic or popularistic rather than science informed. No, absolutely, absolutely. And I think that people are scared because they simply because they don't understand. There was an interesting experiment a few years ago in the US in which people were asked if they fear genetically modified plants for food, so GMOs for food. And a certain proportion of them said that they would never eat a GMO plant or a GMO tomato or whatever. And then the same persons were given an expert course what genetic modified organisms mean and how genetic modifications inserted and which are the benefits, which are the potential risks, how these risks are mitigated, so on and so forth. Well, 2/3 of the person were against GMOs. Then they changed their mind and became pro-GMOs simply because they have been informed what GMOs in that case they meant. So I think it is really a matter of education. I have a cartoon that I often show when I'm asked to speak to the lay public about these problems. The cartoon shows a horse. There is a farm, and there is a horse who runs free and rush away from the farm, and then there is a cowboy going after the horse, but struggling and keeping the pace. And then after several dozen meters, there is the cowboy's wife who struggles to run after the cowboy. And the cartoon is meant to portray science, which is a horse and runs away, and then bioethicists, so the ethical discussion, we try to catch up, and then the last is the law and the jurisdiction and the rules. And this is reality, science gets free, and then bioethicists struggle to stay close, but law comes after. And I think that the decision of potentially dangerous activities must be a decision taken by the whole community at large. So there must be scientists but there must be bioethicists, there must be patient engagement in decision related to medicine. There must be philosophers, sociologists, and obviously people who knows of law. So it is a collective decision. The problem is that these components of the society, besides science, are always slow to react. There have been recently two examples in which were the scientists themselves that imposed or proposed to impose to themselves regulations because the jurisdiction was not quick, was not reacting quickly. One was now there is the possibility of generating embryos using the same technologies that convert any cell type into an embryonic stem cell. Well, any embryonic stem cells can be converted into an embryo without going through an egg cell fertilized by a sperm cell. Now, there is a law that prevents regular embryos, human embryos, to be cultured more than 14 weeks for obvious reasons in the laboratory. But these embryos obtained without normal fertilization are completely unregulated. So one in principle could obtain a human being with this technology, it would be perfectly lawful. And this is the reason why the scientists in this area themselves proposed a code of conduct by which these embryos are also regulated. But this is just to fulfill the lack of regulation for legislator. So this is an example where it is scientists who somehow fill up the gap, but it should be the all components of the society who should take these decisions.